Detox the roots and heal this pain.
Data and the dirt, that's the way. Science and the soil, every day. Let's dig in.
All right, y'all. Welcome to this week's edition of Data in the Dirt. And we didn't do a lot of promo for this. So those of you that are on live, we appreciate the last minute because we were working on this last night and realized this is information we need to get out as soon as possible. So guys, I am Julie. I am the data. I'm a stage 4 cancer survivor who beat the odds using data. Data gangsters don't hope or wish. We know, and we know how to use data to give ourselves an advantage.
And I'm Trevor, the Dirt, declared legally disabled at 18. I healed myself with plants. Now I'm a herbalist and supplement formulator, here to separate the snake oil from the science.
We deserve to heal instead of treat disease.
Let's dig in.
So guys, I am so excited for our episode today, and we are going to be sharing with you how to break down the most important data you can have around your cancer journey and understand how to actually do research. So we now have all of these beautiful programs like AI, things like that. So we're going to walk you through what does this look like and how do you begin to actually interpret your data. I truly couldn't be more excited. Trev, anything you'd add to the intro before we start to dig in?
No, let's do it.
Okay, perfect. So, I just want to kind of set the stage from a historical perspective a little bit, because I think it's wildly important for you all to understand that we are just now in 2026 beginning to understand cancer in a whole different way. In the last 10 years, something called next generation sequencing reporting has become the fad in oncology. And this means before 2015, we kind of thought something was just colon cancer. And now we understand that we can begin to look at things so that we understand what is what beyond colon cancer to understand the actual specifics of your cancer cell. So I think this is a really, really important thing for everyone to understand. But I do, before we get into this, just quickly want to say out there for everyone, hey guys, just in case you didn't know, we are not medical advice. Trevor and I, neither one of us are doctors. We are here to break this down as 2 survivors, 2 researchers, 2 people who dug in to figure out how we could do this differently. So I just want to make sure for all of you out there, anything we're telling you, you then need to go and have a discussion with your medical team. So just wanted to put that out there so nobody gets sued from our conversation today.
I am not a doctor, and I don't play one on TV. So
I was going to say sometimes I play one, so
Wow.
Okay, everyone. So let's kind of begin this. And so glad that we can start our session and begin to break down the data that's so important in your cancer journey. And many of you guys have heard my story and that I asked for a test. I didn't even know really what the test meant, but I had to call my oncologist 20 times to get something called a neogenomics report. And I wanted to do this because someone told me that if you knew this data, you could treatment match before you began treatment. And I was like, this is crazy. There's no way we could know if I was chemo resistant before I start chemo. And he's like, oh girl, there is. So I called my oncologist 20 times. They told me they couldn't find my tumor. I had to call the pathology department at my hospital and find my tumor and get them to order this report. And guys, I got this report exactly at 5 o'clock when I started chemotherapy the next day. And I got this report, and when you look at my data, you can see very clearly that I had a KRAS G13D tumor, and I was MSI high. And so I got to work understanding my report and beginning to dig into that. And I had something called PubMed, which we'll introduce you to today. And I started to fast and furious Google anything that was on the report because it was all mumbo jumbo to me. It was a bunch of letters that I didn't know how to make sense of. But I understood very quickly that the chemotherapy that was prescribed to me, a platinum-based chemotherapy, was not likely to work for my type of mutation based on some research that was done in lung cancer. And right away, I recognized it didn't matter where my cancer was. It mattered the type of mutations. That's the magic. That's the key. So this report that I had in front of me was one of the most important pieces of data you could ever imagine. And so I walked into my first chemo with my best attorney friend, and I slid the piece of paper across the desk, and I was like, looks like we're going to need to do immunotherapy. That's going to be a better fit for me. And he kind of laughed at me and said, Julie, I'm the chief of staff. you don't know what you're doing. You're starting chemotherapy. That's the right thing for me. Trust you. Trust me. But guys, as you know, I didn't really trust him. I kind of trusted him. And when he told me that, I started to measure my blood levels outside of the oncologist. And I was able to prove that the NGS testing was correct and the chemotherapy was not the right fit for me after only 2 infusions. So this data is so important to understand. And now we actually have tools that will help us interpret this. So we wanted to spend time today walking through this data. And then I'm going to turn this over to Trevor because his specialty is understanding how to look at research differently so that we really have every advantage in our journey. So guys, I want to take a minute and just show you what NGS testing looks like because I think it's really important for you to understand this. And for those of you who are listening, this could be under the name of Caris or Tempus or Guardant or NeoGenomics. Lots of different companies have this next generation sequencing testing. So this is something that you may have had drawn based on your tumor. And that means they actually send in a biopsy and then they look and measure the sequencing based on your specific piece of tissue. And then, or they can also do this by looking at methylation patterns in your blood. So a NGS test can either be tumor informed or based on your specific DNA, or it can be looking for a broad swath of different cancer types and be looking to see if you have any of those methylation patterns in your blood. So you do not need to get a tissue sample in order to get this data. Number one. Number two, as you can see here, this example gives us a number of different ideas of things that could be valuable. So PIK3CA, FGFR2, TP53, all of these are things that we could potentially leverage in our journey. But the reality is we don't necessarily know which of these are valuable targets or not. So the very first thing I want to encourage everyone out there is if you have your NGS report as part of your electronic medical record, go ahead and pull that up because you're going to get curious as we talk about this today and go ahead and have this piece of data behind us for the conversation. So this is one example of an NGS test, and I certainly have many others that we could potentially share with you, but I think it's really important for you just to understand the truth is this is going to have a whole bunch of letters and numbers that you don't understand and you don't necessarily need to understand right now. We need to make sure that you can understand what to do with these and how to actually research. So guys, I'm going to see if I can do this, and I'm not sure how quickly I'm going to make this happen. I'm going to see if I can actually, go ahead.
Are doctors running the biopsy report on these at the initial diagnosis, the biopsy diagnosis?
So this is something that when I had cancer, the truth is absolutely not. I had to request it, I had to call 20 times. Yep. Now, more doctors are likely to run a tissue sample as soon as they do a biopsy. But that doesn't necessarily mean they're going to do this without you asking. So you definitely want to ask, have they pulled the NGS report? And so, guys, I'm actually going to show you my NGS report. So this is actually from my cancer journey. And you can see here, again, a whole bunch of gobbledygook of numbers and things like this. But you can see when you look at this, at least in 2022, there were no therapies approved for my type of cancer. So that would have scared me right at that moment had I not understood that MSI-high opened me up for immunotherapy. But Trev, knowing what you know, that doesn't scare me as much anymore because I know there's way more than just pharmaceuticals. So would you mind just taking a minute to walk through how you would look at either one of these reports? And I'm happy to show up the other one or keep mine up, whatever you'd like. But I'd like to think about how someone might look at this report and think about some of the things they might want to potentially search.
Yeah. So, well, first off the bat, obviously, it's good to be empowered and to be able to do this yourself. But this is some of the stuff that's going on in the background with naturopaths and some holistic practitioners. And you definitely want to ask them and make sure they're doing stuff like this. But so just so you know, you don't have to discover these treatments completely by yourself, but it's also good to be empowered to do that. So some of the things we'll do is, so say like for Julie's specific cancer type, I personally would start with looking up all the unique variations of markers and genetic variations, stuff like that. And I like to lay them out. And also right here, Julie has the test of which ones she had for her specific tumor type. So using that, you can go on PubMed, which is an American database. I guess it's got European, American, it's got some other countries' research in it. And you can cross-reference and search these terms, and then you're going to get a mix of pathology papers, as well as disease processes papers, as well as pharma research, but you will also find some supplement research and different medical interventions like that. So if you were starting from scratch and just trying, what is there outside of chemo? Say you're just starting at base level, you could start searching through those things. And this does get a little bit deep. So it's not everyone's cup of tea. Just want to point that out. But go ahead.
I think actually, though, I just want to, if it's okay with you, I'm going to see if I can make this work.
Yep.
I think it's important for people to understand what is PubMed. So do you mind just quickly?
Oh, yeah, I apologize. Yep. It's just a government hosted database of published studies. And it's not just medical studies. It's mostly medical studies. There's a small amount of some like ecology studies and different things like that. But obviously we're looking for peer reviewed studies. We're looking for studies published in peer reviewed journals, rather, is what I meant. So that means someone else has had eyes on the studies before it's published to the public to verify that it's at least indicative of a pattern or anyways, it's been put through someone else's filter in that field is the thing I'm trying to say. And they decline publishing things that are disagreed with or things that are a little off, which can be good. And sometimes stuff gets declined too. Anyways, it's a mix. But okay. So PubMed is one of the initial databases you go search for any medical research. And so for like in this case, sorry, I have one for a different case in front of me. So I'm just going to start talking on that one. Is that all right?
Yeah, of course. That's what I was going to say. Let me put that up so that people can see what that case,
Okay. Yeah. So we're doing research on a case with a signet ring cell morphology. And so it's a signet ring and it's mucinous and it's adenocarcinoma. And then of course, location matters as well. So anyways, I created a cross-reference list of all the search terms associated with that diagnosis and with those different disease diagnoses I just told you. So I came up with a list of specific cell morphology related to signet ring. So it might be like E-cadherin is a big thing where it's turned off. And so the cells don't stick together. And so they kind of tend to float off and then deposit in other places in the body and start new tumors. So, and that's a target that is both targeted by pharmaceuticals as well as there are supplements that target that. So let's say you typed in PubMed E-cadherin, or you might type the shorthand, which like might show up on Julie's report if she had this, it would be like CDH1. And so that's E-cadherin 1. So you would type that in to PubMed, search it, and what's going to happen is you're going to get a lot of papers. You're going to get, like I said, you're going to get papers on that mechanism, that specific morphology.
Let's do this real fast. Tell me what to search. Let me just remove this from. And I was going to say, let's actually show people real time. So what's something you would? Yeah.
Just type E and then cadherin. E and then second word cadherin. Yeah. Or you got it. I didn't have to spell it well. Good job.
E-cadherin and what? Anything?
Well, just type that. So search that, and I just want to show what comes up. And then you can get into deeper search patterns. But so if you look at this, you're going to get studies. You're going to get like disease pathology. You're going to get research studies. You're going to get everything on it. And how many papers do we have? How much does it say up top?
Up top, 4,615.
Yeah, so quite a bit of papers on this topic. And most of these are not going to be on medical interventions. So what you would do further is you can do E-cadherin and you can do plus, or you can drop down the detailed search option, and you can add in qualifier terms. So a qualifier term might be like E-cadherin plus inhibitor, and then you search that. And what that would bring up is anything being studied that inhibits E-cadherin. And so there you go. And I'm not saying all these are going to be useful to you. A lot of these are going to be research, chemical studies and stuff like that. But for someone like me, I'm digging through looking for supplements, compounds from plants, compounds related to plants, anything like that. And then if I get a hit on something, like, oh, look, I just found E-cadherin. I apologize. I can't think of one off the top of my head. I don't want to misstate it. Well, anyways, let's just give an example. I say I find herb A just happens to affect E-cadherin. Then I would start searching that herb and E-cadherin. or it may be a compound from an herb, and I would start searching through it and looking for any studies on that. And then what you're going to start assessing is the quality or level of research on it. So different levels might be, you might have an in vitro, which means it's a cell study. That means
let's take a peek at that. So let's just pull up one and let's walk through what you would look at to see if it's good or not good. So anything in here that you think would be valuable for us to take a peek at, Trev?
Oh yeah, let's see. I should have cheated and had this preloaded, but I didn't do that.
What about this?
Let me pull up. Sorry, I got so much going on. I can't remember one off the top of my head. Because I find them, I do the research review, and then I throw it in the file of potential interventions. All right, I'm doing it on my end so I can pull up something quicker.
Do you want to share your screen? Do you know how to share your screen?
I don't. You're gonna have to show me. I apologize. Okay, so like a good one is okay, there you go. Oh, it's an easy one. Duh. Oh my gosh, that should have come straight to my mind. Is quercetin. Quercetin interacts with that, with E-cadherin. And so now you would do that, and E-cadherin, you can do and or a plus sign. Exactly, perfect. And then you're going to look for cross-qualifier papers on that. And quercetin's a pretty well-studied plant constituent as an antioxidant. And so you would go through these. And me, like 20 years ago, I would go through and read the abstracts on all these. The abstract is a quick summation of the paper. And if I felt like it was directly relevant to the actions on E-cadherin, then I would start reading those and I would start cataloging them. And I'd be like, okay, we have 5 papers done on cells in a Petri dish. That's like, other than computer modeling, it's like the lowest level of evidence. Because guess what? We're not just cells. We're a whole organism. We're a whole city of cells in our body, right? And they all work in different ways and have different actions. So when you put a substance on a cell in a Petri dish, it's nice to see if it does something and it can indicate something, but it's not the highest level of evidence. So
hold on. Can we take just a minute on that? Because I think that's a really important thing for people to understand.
Yep.
So some evidence, as Trevor just said, is in a Petri dish, which means they take some of your cells and they see if something can help or hurt it based in a Petri dish. This is what tests like the RGCC do. Right. It does not mean that they will work for you. This is what many of the tests like that will do. Yep. It just means that in a Petri dish, there was some sort of reaction. And so as you just heard from Trevor, that's the lowest level of research.
Yeah.
So sorry, keep going, Trevor. I just wanted to make sure.
No, it's great. Yeah, it's a great explanation. Yeah. Yeah. Like the lower level below that is computer modeling, where you try and decide if things work based on computer info. And that used to have like 99.9% miss rate. I suspect it's getting better with AI. So that might become a better level of starting. Anyways, so cell line studies or Petri dish studies in vitro, pretty much the lowest level, but it gives us a place to start. If an action happens, great, let's study it in a living model. So then you get into animal studies. And so then we'll go through and be like, oh, here's some quercetin studies with animals. And you can kind of assess effects differently. We have in science, it's kind of laid out what animals have what in common with us. So you might see some rat studies, or you might have higher elevation animal studies. The issue is we don't love animal studies because they have to kill them at the end to measure the effect. So it's not wonderful, but they just still do it for some things. And if that data is there, we'll take it in. But and then the next level, obviously, is going to be some human studies, like human research trials. Or even case studies can be useful. Case studies are a little bit lower level because it might just be one client, or it might be like 10 patients of a doctor, or it might be like some smaller thing that assesses a trend they believe they're seeing. Anyway, so we take all those things in. They're all different weights of evidence, and we take them all in. And I want to point out an important thing with animals. This is a common mistake people make. If you read an animal study for a supplement, you cannot convert the measurement. The amount of the supplement used in animals does not equal the amount used in humans, like ever. It doesn't work that way. So a very common mistake is to be, oh, they give the mice, or they gave I don't know what animals they use. They gave the monkeys this. And then you try to use that conversion for you. That's not, it does not work that way. Yep.
So I just want to make sure people can visualize this. At the bottom, it's the computer type of measurement. It's the computer analytics. Next, we would look at Petri dish studies. So these are what he's called a cell line. So that can be the same word. The next would be animal studies, which is could be rats, it could be monkeys, something like that. The next is when we get into our clinical trials. And there are 3 stages of clinical trials. So can you walk us through each of those 3 stages of clinical trials, Trev?
Well, yeah. I mean, a lot of the times you're going to see more. So there's a base level that is case studies, like I mentioned, where a doctor might write a specific study on something they saw in one client or one patient of theirs. So I just want to, or maybe like a couple patients in their clinic. Oh, we're seeing this trend. So I want to point that out. And then ask your question again. Sorry, levels of what?
I was going to say the clinical trials. I want people to understand it goes from a computer study to a petri dish study to an animal study. As we get into human studies, that's what we call a clinical trial. And there are clinical trials to see. The first clinical trial 1 would be, does this work for humans?
Right.
Number 2 was when we started to get to dosage. So we walk us through kind of how the levels of clinical trials, Trev?
Yeah, totally. All right. So sorry. So we got, okay. So you laid it out. You got preclinical stuff like the in vitro, the cell line. Then you got animal. Then you have the case reports or case study series. Then you have what they call like a retrospective cohort study. So you're kind of looking back at patients. This is like post-treatment, and you're trying to compare outcomes. So it's like a type of study. Then you get into, let's see, like, I guess retrospective studies. No, that'd be ongoing. I'm sorry. I'm just trying to think through the different variations. Because we did a clinical observation study. That was the breast cancer study I was on at Fred Hutch. It was a translational outcome study. So that was active monitoring. We matched people to equal cohorts. And then we tracked one standard of care, the MD standard of care, with the naturopathic Fabno standard of care for breast cancer. And everyone was matched very specifically on HER2/neu and ER/PR status and all that, and staging. And so that was a translational outcome study, which is very useful. It was zoomed out. I think it was, I can't remember, maybe like 3,500 people in that study, 4,000, something like that. Then you get into the different levels of clinical trials. And like phase 1 is usually just safety and dosing, like figuring out what dose works. So if something's in a phase 1 trial, especially when it's a supplement or an herb extract, usually pretty safe. It depends. Everything can potentially be dangerous. But they're figuring out toxicology in a phase 1. And dose it. So in a pharmaceutical trial, that's really early. I would always be cautious, depending on what they're studying, because it's early data. Then you get into phase 2, which is a little bit bigger. You're trying to characterize the side effects and things like that. And then phase 3 is the exciting part with drugs, and some supplements make it to this level, especially if they're in like an investigational new drug process to become a prescription, which they do that with plants. And that's where you get into like the randomized controlled studies with people. Like that'd be considered like the gold standard, where you're like giving one part of the study, you're giving a placebo, and then one group in the study is getting the treatment. And then they have this ethical thing where if the treatment shows like an effect, I don't know how they measure it, but an impressive effect, they have a crossover thing where they will start giving the effective thing to the people that were getting the placebo. In some studies, I haven't worked on something like that, so I can't quite qualify how they're doing that. But
I think it's so important. The number one thing when you look at a piece of research is this a meta-analysis or metadata study where they're bringing together a bunch of studies into one?
That's where I was going next. Yeah.
Okay.
That's the next level. That's the next level of study is metadata. Sorry, and then I'm done. Yeah.
Okay.
But that's exactly right. Yeah. Metadata is like an umbrella study. You are literally using data scientists and then scientists that are honed in on the specific area of study, and you are pulling every paper on the topic, and then you got to go through and qualify the papers because there's all kinds of errors to make in research. I'm from the herb world, so I always go straight to an herb example. But like, say you didn't identify the herb. You'd be shocked how many studies they didn't even bother to identify that they had the correct substance. They just bought it at the store or something and then did it in the study, used it in the study. Or say they didn't look at how the herb works. Say there's a great study. I mean, it was a really well-intentioned study for radiation therapy side effects. I can't remember the university that did it. There's a hospital on the East Coast. Anyways, but they did it for radiation therapy, really well-intentioned, turmeric, and they gave it in a tea with nothing in the tea, and they said it didn't work. Everything in turmeric is fat soluble, or the majority, everything that's like exciting for radiation therapy and helping with side effects in turmeric is fat soluble, so it doesn't extract in tea. And then they said in the study it didn't work because they didn't look at the solubility or the traditional uses.
Okay, so I just want to take a minute because what you just said, a lot of people that were listening probably won't know that some herbs are water soluble and some herbs are fat soluble.
Yeah.
So what we just described is a research study that said this herb is ineffective But really what was ineffective was the study design, because they didn't ensure that the fat soluble vitamin was given with something that was fat soluble alongside of it. So this is where it's so important. People will say certain herbs don't work, and you're like, well, they did 1/10 of the dose you should get, or they did 400,000 times the dose you should get, or they used the wrong part of the plant, or they didn't mix it with fat soluble. So this is where the research, you got to actually understand what you're looking at. You just can't take what AI brings back to you and take it as gospel. So sorry, Trev, I just wanted to make that quick.
No, that's great. It's so good. It's so important. And that study specifically, like I talked to one of the investigators in it, and it was really well intentioned. They were seeing good effects with turmeric with their patients going into radiation therapy, and they wanted to do a study on it. They're like, oh, this is great. These people are having decreased side effects. And then, they didn't do the study properly because they're not whatever. They just weren't like plant chemists or pharmacologists or whatever. So it wasn't even bad intentions. Anyways, I just want to point that out because it wasn't even conspiratorial. It wasn't conspiratorial. It was just like, it was an honest mistake. Yeah.
And this is the piece of why I think it's so important for us to make sure we're working with a qualified herbalist or naturopathic oncologist that actually understands the research and understands what this means. So I just want to underscore for the professional people like you, Trev, to say this is why you go to talk to someone, because they understand what's real. And there's been tons and tons of research out there about, oh, you can't use vitamin C or antioxidants. That's not true. There's a whole reality of like, you got to understand how to dig into this to understand where's the cream from the crop.
So and it's well established. Yeah, there's like a well established safety profile and like a bunch of studies. Okay, maybe not a bunch in the research, but more than 10 studies on vitamin C being used during chemotherapy, being used right before, being used on the day of, being used on all the different windows. And wonderful safety study. Anyways. Sorry, but it's really important to lay it out. In AI, we're at this very cool intersection, like you said, where you can use AI. And we even have AI specific to research, where they've really honed in. It does tend to err on the side of the FDA recommends. It does tend to err on that, but you can parse through it as long as you know how to, what do you call the terms you tell AI? What is it called?
Prompting.
Yes, they're prompting. Yes. Oh, yes. There we go. As long as you are prompting correctly and you have some subject matter knowledge, you can prompt through the issues and be like, yes, but how many people did that in the study? Did they identify this in the study? Did they do this? And then even like Cite AI, which tends to lean towards the FDA, it'll break down the research articles, but it tends to lean towards this doesn't probably do anything. It will answer those correctly if you prompt correctly. So it is cool. We're at a very cool intersection. And like the project me and Julie are working on right now, I'm doing this metadata. I mean, it's effectively kind of a metadata study. It's a little bit different, but I'm searching for all those search terms for specific clients, like a cancer type, all the different genetic markers and cell morphology. I don't know why I keep mispronouncing that word. But we're searching all those terms, and then I'm using qualifier terms. So we kind of mentioned that earlier, but it's to help bring back just the research terms. And right now, you cannot do a project like this with just AI. AI will miss things when it's scraping through the database, but we're getting close. And so I'm setting up a thing with people to actually help me pull the data in the old school way with data specialists. Me and Julie are like setting up people to do that. And then we're going to use that. And then we're still going to use AI to help sort through the material that's going to be incoming. And then you still have to lay eyes on things though. So you can use it for sorting. You just can't use it for the final determination. Exactly.
Okay. So let's take a minute back and let's kind of,
okay,
so our listeners can follow through us. So guys, the number one thing you want to do is to pull your NGS report, and the NGS report could be titled Caris, Neogenomics, Guardant, etc. This is the report that defines the mutations of your cancer. Not all doctors will upload it to your MyChart or Epic or whatever it is. So if not, ask your doctor for an NGS test. And if they have not done it, ask them to do it right away to help you with treatment matching. So number one is get your NGS report and understand and familiarize yourself with the targets. Yep. When we think about NGS or targets for this report that I have on the sample right now, Trevor, name the one, when you look at this row on this, what's on this sheet, what are the things that you would pick out to put in a search? Okay.
So right here, so right off the bat, you can use Pika 3CA. And then Pika 3CA, I'm trying to see, because here's the deal. So you'll look at a search term like that, Pika 3CA, and very important target if you can hit it. And there are some things to affect it that like naturopaths and some of the holistic oncologists are using. I'm pulling it right now because what happens is you then need to search on the side. Okay, here we go. So you need to search on the side any associated search terms with it, because sometimes in a research paper, they might refer to it by another term. But to the research scientists who are in that world, they're like, oh yeah, that's Pika, or a Pika 3CA. So for this one specifically, I had to flip to it because obviously I had to look these things up. E542K is a associated base term for that mechanism and marker.
E542K, you said?
Yeah, you don't even have to do an and on it. Yeah, I believe it's just Pika. Yeah.
Okay.
You can do an and, it doesn't matter. But
Okay.
when you're doing it, you're on site. Yeah, okay, yeah, do that. Yeah. I'm still learning Site too. I mean, it's pretty intuitive, but I'm still learning it.
Here's what Site's a program that I use quite a bit, guys, and it helps me kind of just quickly rip through some research to see what I need to then go in and actually assess, as Trevor has mentioned. So it makes life way easier than using PubMed, as I had to do when I was diagnosed. Site is a program that can look at almost any medical journal that's in Western hemisphere. So this does not include, as Trevor mentioned, some of the things in China, Russia, India, etc. But this is going to give you a very broad spectrum way to look at research, and it will give you back information in English. So it is a very valuable tool for those of us that don't have Trevor's training in brain.
Do we want to? I actually didn't mention that yet. You've just heard me talk about it so many times on this project. I didn't mention the foreign database thing. Do you want me to mention it?
I think it's just important to say, guys, this is going to be a pretty broad swath, but it's not everything. So I think we can just go forward with that.
Yeah, great. Yeah. But as far as tools, actionable tools, Site AI really makes this very accessible to the average person. Let's just state that right now. And
we're not being paid by Site AI either. I just we actually pay Site AI. So
that's right. Yes. Yeah.
Yes.
But we're waiting for our sponsorship in the mail, right? No, I'm just kidding. But I'm saying, so the old school way was to go to PubMed and comb through that. You don't quite have to do that. I'm still a little not quite there on trust yet, but it is, Site AI is amazing. And I would say jump to Site AI first for your basic person trying to do this, because jumping into PubMed, you can totally learn it. But it is, it's a different world if you haven't used it before. And Site AI, it is pretty good. Yeah.
Okay, great. So you were going to say that the two terms.
Okay, yeah. For this one, just type that and type. Let's see, because Site AI, you can. It's AI. You can speak to it in prompts, which is amazing. Sorry, I'm repeating that to myself. So, so you can just say, please research treatments or medical interventions affecting this or something like that. I don't know, or research on things affecting these markers. Yep.
Okay, great. And then I will just hit go. Oh, sorry, guys. I messed something up. Someone asked me to put this link, and I realized it's not flipping over to Facebook, so I'm going to try and do that real at the same time.
Mm-hmm. Oh, we probably could have honed it in a little better and done a Pika direct supplement that we know.
So the thing I really like about Site, so I'll just kind of take a minute to explain, is it goes through all kinds of research, and then it shows you at the top. It then will go back and fact check some things. But guys, it's important for you to understand any AI can hallucinate a little bit. So I'm never going to accept one AI's perspective as gospel. I have memberships, frankly, myself, to Site, to Claude, to Perplexity, all kinds of different things, because I fact check them against each other. So I think this is, as you can see here, it will go ahead and fact check as much as you can in Site. And I also really like Site because it'll allow you to see the article it's referencing in its collection. So it's not behind a paywall, because I pay for Site. So there's a lot of good advantages of using this particular technology. Yeah,
it's really good. I mean, so like I felt there would be some minor errors with Chat GPT, or at least there were. I haven't used it in about a year. Claude, I use more, and there's less and less errors. Although they just did some edit recently, and that's flagging a lot of medical research stuff. It's weird. It just started happening to me yesterday. But Site AI, the point of Site AI, it is made by medical researchers for medical researchers It's specifically targeted to that world. So they've really done good on filtering out. And I mean, I haven't really seen a hallucination on site AI that I can recall. I've definitely had them in Chat GPT and Claude before. I mean, a year ago, I got like fake citations studies that didn't even exist. They were telling me. And when I went to read them, I guess people don't click the links. So when I went to read them, they weren't real. But that's happening less and less.
So it's getting much, much, much better.
Yeah, but Site AI specifically for research is great. Let's see.
Okay, so now you go through here.
Sorry, I could have front-loaded examples for us. I just didn't think about it.
That's okay. So anything here that you'd recommend us looking at or showing so that we can actually look to see if it's a trial, a phase 1, phase 3, animal study, cell line, etc.?
Yeah, well, I mean, I just go through and I read all this. Yeah. So I would start reading through all these. And what my mind is looking for is, obviously, I'm looking for any intervention. So anything they're using that is affecting change on this target. And if there's like a pharmaceutical or a clinical trial, I denote it. I read about it and denote it because it can be of interest. And then, of course, I'm always looking for complementary items like supplements, herbs, could be IV treatments, could be a number of different interventions that are complementary to the MD treatment of care.
Okay.
If that makes sense.
So let's actually
look. Why don't we do this?
Yeah.
Okay. Yeah. I was going to say, do you want to jump back and do you want to pepper it with a supplement I know that has an effect or has some research on it for Pika?
I'm just wondering if we can look and see, like, does this tell you anything about how the quality of this research from this?
Yeah. Stop right there and I'll read this real quick.
Okay.
Yep. And scroll back up to the top to the title. So that was just the explanation of the disease.
Okay, so this isn't going to help us.
Oh, yeah. So it's just characterizing. That is just character. So that was a great example of what you will get a lot of. And that is a paper characterizing the disease or that specific genetic marker or mechanism.
So here's one that's actually about a study.
So, and those are great to help you understand, but we would definitely, that's not going to have interventions mentioned in it. And people aren't going to be using, oh, here's an intervention. People aren't going to be, I'm just using that word just so you understand. So this looks like a great paper. It's not, it doesn't, I don't know if it has direct actionable intel. So I would be reading through this and then I'd be looking up these pharmaceutical drugs and be like, oh, interesting, looking at those. This isn't going to be something you would be using outside of care, but this can help determine, at least bring questions to your doctor, because these are pharmaceutical drugs mentioned in the study. And so you'll be searching, are these in clinical trials? Are these available pharmaceutical drugs as you're reading about it? And then for someone like me, I'm also looking at the target, what it's doing. Oh, it's inhibiting, it's a TORC inhibitor, or it's a WEE1. And I'd be reading through, looking at this, kind of denoting in my brain, oh, interesting. And in my mind, I'm mapping out where that's hitting on the map of targets for the specific cancer case, like for the specific cancer types that I've already mapped out. So I don't know how useful that is for other people to be doing that, but that's just how I have to do that to start map it out. And then as I start to dig into the supplement and other intervention world, I guess we call it the holistic intervention world or complementary intervention. I don't know. Anyways, then I start looking at any research that's affecting those targets.
Okay.
So this appears to be looking at multiple clinical trial drugs, and a few of those may have been on market. But for me, when I'm digging into a case, every little bit of understanding the disease pathology and treatment targets and what they're already looking at are helpful. And then, do you want to go back to the search bar and let's add in a supplement that I know had some studies on it?
Absolutely.
All right. Let's see. Let's fisetin. Let's look up fisetin in that. I believe that was a Pika one.
And Trev, we did get a question, I'm not sure if you can see the comments, that asked if there's a way to use this to understand the metabolic function or fuel sources. So let us know if there's a way to research.
Yeah. Well, I know they can from the actual biopsy, or if it's a removal from the pathology. you can test them to assess what the tumor is feeding on, right?
How did you? No, I don't know what you're talking
okay, sorry. What I'm referring to is whether it's
We're
like
wondering if with research, if there's a way to use research terms to find out what the metabolic function is or the fuel sources are for the cancer that we've had on the NGS test. But I think what you were just saying,
I got you.
But I think what you were just saying is that you can actually just look at the NGS report and understand.
Yeah. Well, I know that they can tell that information from it, or from the what's the imaging dye they use to tell its glucose metabolism feeding rate?
The radiologist.
Yeah, you know how they'll do the glucose dye, and it tells you how fast the tumor is metabolizing sugar. That imaging is not a biopsy, and that can actually tell you. Oh, they can denote if it's like a lot of cancers are glucose metabolizers. They're sugar hogs, and then some rare types are feeding more on amino acids, proteins, and so they can assess that because those won't be metabolizing the sugar. Okay. So that's not even a biopsy report yet. But I don't know if that exactly answered the question. But yes, that data is available in these research studies. So I'm just going to go. My head's just in the case that I'm on. I apologize. So I'm going to go back to that. With the signet ring, I was looking at different articles on that, and there was an entire article laying out how it primarily fed on amino acids and proteins, and a couple specific amino acids, and which was of importance here because I want to say it was I can't remember the percent, but there was published data showing a very low percent of glycolysis feeding on glucose. And so we've talked about taking.
I'm so lost with what you just said. Sorry, Trev. Can you just recap what you just said? I just got lost in the glucolysis things. That was still on. We were still talking about it, figuring out how the metabolic function of a fuel source using the data, right?
Yeah. So I was just giving an example of where I found that information. Yeah.
Okay.
So I was searching about signet ring and just information on. I was curious about its food source, what it metabolizes, stuff like that. And there was a published study, I think it was in 2022, and specific to signet ring. And there's this info on other cancer types. But signet ring had a whole paper on it because it largely metabolized proteins and amino acids as a food source, and to a lesser degree, glucose. But still the Warburg effect still did eat glucose, right? But that indicates, oh wow. So now we've talked about that newer paper on glutathione, and how some cancer types are able to disassemble it and use it as a food source, the amino acids in it. Well, signet ring would be a cancer type that could do that because it has a predilection to feeding on amino acids. But the question that we was asked was, how do we find out the metabolic function or fuel source? So what you just said, I just want to make sure that we're kind of wrapping this up for the question, that should be part of the report. So where did you find that for signet ring? So that's what I'm trying to understand. Okay, so I found that on PubMed. There was a single study on signet ring and its food source. Yeah. So you could search then signet ring or whatever the qualifier is and food source, and that might give you information. So that would be the right search term, right? Right. Probably if you're going to use more generalized terms, I would prompt it into Cite AI. Do you want to try it right now? Well, I was going to say, do you want to do anything else with the phycetin while we're here? Or no, I was just giving an example of that was one I had done a study report on with the Pica marker. We were going to explain how we understand if someone's a phase 1 or phase 3 trial, whether this is a human trial or petri dish. We still haven't explained how we do that. So let's pick any study you want, whatever you want, and I'll pull it up. Yeah. I mean, I'm literally reading all these studies to determine that. I apologize. I'm having trouble answering the question. I need to understand how we would look, where we would look in a study to understand how we would quantify that. Where it is in the study process. Yeah. Where in the research article, how would I know if this is a phase 1 trial, whether this is a clinical trial, whether this is a... Yeah, it's going to tell you at the beginning of the article. If you're reading a clinical trial paper, it's going to basically state that it is a clinical trial paper. It's going to say a phase 1 study of or a safety and efficacy study of a compound. It's going to say it in the abstract. Yeah. Okay. A lot of the things, I'm sorry, I'm having trouble answering because I'm basically getting some of these answers from reading all the papers about a topic. And then that's where I'm trying to understand. When you look at a research study, what do you look at to understand if it was in a petri dish or in a human? Oh yeah, it will state it. It's going to state it. If it's in a petri dish, it will say in vitro. And then go ahead. Where?
That's what I'm trying to look at, one research study where we can actually find that so we can teach people this.
Oh, okay, yeah. Did you find a study on fisetin or one of its specifics? I mean, so in the abstract, it's going to tell you, it's going to directly say, this is an in vitro study of the effects on fisetin on so-and-so cell line.
So here's the abstract. Let's look.
Yeah.
This is
low PTN levels. Oh. So go back up to the title again. Low level PTN levels and PICA3 mutations predict resistance to neoadjuvant patients with HER2 overexpression breast cancer. Okay. All right. All right. So they're studying that. Okay. The resistance to that. Okay. All right. Got it. I mean, the answer is you read the paper and it says it in there, or Cite AI, you can ask it and it will.
Where in the paper would it show it? That's what my question is. So where would I look? Can you say?
So it may say it in the abstract, or if not, you're going to go down to research methods. So purpose, patient methods. So it could be in methods, or
so this is on patients. So we know it's a 59 to 46 patients. So we just net right now in the very top, we could say right away, because it talks about how the samples are from patients. This is number one. It's not a computer study. It's not a Petri. It's not an animal study. It's got to be at least some sort of human study at this point. So we know it's not a Petri dish, right?
Yeah, there you go, under patient methods, 12-week neoadjuvant clinical trial.
Okay, perfect. So right here, you would look just in the abstract to understand more about the actual study.
Yeah.
Okay.
I apologize. Usually, it's the reason I didn't. It's usually in the abstract, but sometimes they don't say. So sometimes then you go to the research methods, or in that section, they called it patient and methods. And sometimes you have to go to the results. And then sometimes you're emailing the author to be like, hey, what was going on here? It's not always clean. Yeah.
Okay. So we have.
I just jumped through those things trying to figure it out. I apologize. Yeah. That's why it's hard. It was hard to answer. Yeah.
So I think, because I think we just confused a lot of people. So I want to kind of recap this and say, okay, so I think that what we can do is use technology, and there's multiple different technologies out there you can leverage. Cite is one, Perplexity is another. So I just want to mention Perplexity really quickly.
Yeah.
That is another software where you can come in here and you can actually do different types of research. So I would certainly recommend that you do this. Right here, you would go to your connectors page and you'd move from web to academic. And now you can ask any question, just like we just did on Cite. Each of these are about $20 a month, so they're not free, unfortunately. So, but I certainly use Cite and Perplexity quite a bit. Either one can give you journal articles. So I think if you have a subscription to either one, this is how you would do it. The idea is that you would go to your NGS report. Let me just quickly flash that so that you remember what that looks like.
Yep.
You would go to your NGS report and pick any one of these factors. So PIK3CA is the first one we thought about. And then you would simply say, is there a treatment impacting PIK3CA? And that will start to surface this research article so that you will understand what you can do. So I think then we would suggest looking at the abstract. The abstract should say, is this an animal study? Is this a human study? So that's the process we would say is start with your report, go to your technology like a Perplexity or like a Cite, use your report to pick a term and pick something like treatments impacting this term. And you would do that for every single combination that's on your NGS report.
Yep. And now I want to flash forward to someone that I read through the results that was one of your clients, Julie. And they actually plugged in these elements, right? They plugged these elements into basically a case into a file. I don't know if they're using Claude or not. But they created like a file, plugged these elements in, basically laid out their case, and then started to search through things that could potentially affect their different aspects.
That's exactly right. So I love this story because I was just looking for some rare survivor stories. And I had an interview with someone that I'd spoken with about 2 years ago randomly. He read my book. I'd shared a little bit about that. He then took the knowledge that he had from me and his doctor and started to use ChatGPT. I would also suggest you use things like Veda Life. That's going to be a big help for you. But what he did was he uploaded his NGS report. And then he said, explain to me any sort of treatment that could work, taking away FDA requirements. So that's where he wanted to begin to understand when you break this down, what else is out there from an herb perspective, from a lifestyle perspective, from a diet perspective? Because remember, the FDA does not look at herbs. So anything that has to be FDA approved, it's not going to look at herbs because herbs are never FDA approved. So this is the point of you need to take that removal off because they will only try and respond to you with things that are FDA approved. So you would say, upload your NGS report. You would then say, provide to me treatment options or non-FDA approved treatment options and prioritize those based on what would be the best fit for my type of cancer.
And these days, like I just said with Claude, I was writing a research paper. I was actually just using it to collate research that I had already written and was collating it because I'm just like spitting it out. And it blew up a red flag. So I had to denote, this is for research. I'm not using this to treat. Anyways, it's kind of bizarre. I didn't realize that was happening. Things were getting flagged with medical stuff. Do you know anything about that, Julie? Have you had that happen?
It's happening more and more.
It's unfortunate. Yeah.
Yeah, it's unfortunate.
Yeah. But anyways, I was able to prompt around it. I just had to put a context to it. It was annoying because I was just trying to edit a paper, but yeah.
Okay, so I know we went through a lot. We've had about 50 minutes of live so far. So I just want to recap this and say, number one, none of this matters if you don't have the data on your tumor. It is very important for you to understand the exact mutations of the tumor that's in your body and not just treat it as if it's colon cancer or pancreatic cancer or prostate cancer. Frankly, that doesn't matter. I need to know the specifics of if you are a KRAS G13D tumor or if you have a PIK3CA mutation. Each one of those mutations unlocks a different key, a different door, a lock that a treatment can unlock to help treat your cancer. So it gives you a way more at bats, to use a sports analogy, so that you can understand ways you can treat your cancer. Now, when you go to your doctor and you get this report, they're going to tell you what the druggable mutations are. But as we know, there's more than just pharmaceuticals we can use in our healing toolbox. So we want to understand even the mutations that aren't druggable because they may have an herb, a lifestyle, a diet, etc., that could potentially impact them. So you want to look at all of your mutations, not just the ones that are listed as druggable on your NGS report. So NGS is going to be one of the most valuable tools. That's a report by NeoGenomics, Caris, Tempus, Guardant. Make sure you have that as part of your care strategy. As soon as you have these reports, then you would leverage a technology, and that could be Scite, that could be Perplexity, something like that, to begin to do your own research. But guys, here's a little bit of trick as well. If you use technology like radicalhealth.ai, you could go in there and connect your electronic medical records, and they do all of this for you. So if you mention my name, they'll also give you a month free. So please make sure if you go into Radical Health AI, you mention Julie Stevens, because that will make sure your first month is refunded to you. But I think the important parts here is you can do a ton of research based on the mutations using resources like Scite, Perplexity, PubMed, and you can get all these journal articles and begin to understand what they mean. As you assess the journal articles, you want to look, one, is this a computer analysis? Is this cell line or petri dish? Is this an animal study? Is this a human study? Is it a phase 1, 2, or 3 study? Those are going to teach you how serious to take this research. If it's a phase 3 clinical trial, I'm going to take it a lot more serious than if it's a cell line or petri dish. So these are some of the tips and tricks you've learned today. Anything else, Trevor, you'd mentioned to help us wrap up our episode that are really critical learning points for our audience?
Yeah. So a lot of the herbs are going to have some low-level studies, petri dish, maybe an animal, and hopefully a few human studies. But you're going to bundle that together as your body of evidence, and then you're going to look at that supplement, and then you have to look at safety review information. And you're going to need to go over this. This is, unfortunately, at that point, you could bring it to a doctor, and they would just be like, well, I don't know. And they're not going to be necessarily used to assessing safety data. This is where I really do like herbalists and naturopaths and some of the holistic oncologists. But like, let's say quercetin. Quercetin very mildly affects blood clotting. And so if you're on a blood thinner and you're getting chemo and you have these other factors, you can safely use quercetin, but you might say you're on like a blood thinner. Eliquis is a very common one. And so you would just have to potentially time your dosing, and you would still speak to your doctor about it. And this is where it is nice to have someone who's used to assessing these things in active cases, because it lets you feel a little better, because that warning is real, but it's not as scary as you think, as long as you're using it safely as far as timing goes and dosing goes.
For sure.
Yep. I just wanted to point out that safety element. Yep.
Yeah. Okay, great. I hope for all of our listeners today, this has helped you understand A, why you want this particular data, B, how this data can unlock different treatment pathways, and C, how you can use this information to empower yourself to become an informed consumer of healthcare. Guys, all we want is for everyone out there to become an informed consumer of healthcare. We know more about the car we buy, the microwave we buy, the toilet paper we buy, than we do about the cancer treatment we receive. And that ended the moment I reached no evidence of disease, because it's been my mission since that moment for everyone to understand how to cancer and heal smarter. So I hope today's episode of Data in the Dirt has been wildly helpful to help you begin to understand this data and what you can do about it. Trev, anything else you'd mention as we begin to wrap up?
No, that's it. Yeah. Keep searching, keep researching. Don't let me scare you off from it. Sorry, I didn't describe it very cleanly.
I was going to say, I think you described it from a researcher's brain, but we got to describe it from a patient's brain, which means if you feel like this is too much for you, hire someone like Trevor, an herbalist, someone like who's an herbalist, someone who is a naturopathic oncologist to guide your strategy. Guys, this is really hard. So if this is too much for you after you watch this, hit the easy button and hire someone to do it for you.
And let me point this out. The direction I'm going at it from, looking for things to affect those markers, is the most complicated direction to come from. The other direction is to have your preloaded information, like Julie talked about. You already have your information from your report, your NGS report and all that. And now you can look up treatments and come at it the other direction. You will find that to be much simpler if you're trying to assess different things to add in, different supplements, and see, oh, does this have an effect on my specific profile? And then go through the body of evidence that way. I think you're going to find it less complicated than the way I just described to come at it. Awesome. I'm coming at it completely opposite way than probably you would want to. Yeah. Does that make sense?
I mean, I want to teach everyone all the ways. So yeah, I appreciate you so much and appreciate you helping to work through how you would do this research. I want to encourage everyone out there, do the research. Don't just walk in and think I've got colon cancer. Know you've got a KRAS G13D tumor. Know if you're MSI high. Know what your PD-L1 score is. Know your data, because knowing that helps you make sure if something's not working, you can fail fast and pivot, and make sure you can match to the correct treatment right from the very beginning and avoid all kinds of suffering.
Yeah.
So thank you so much for everyone listening. We're so grateful for you. Hope this helps. And we will be back in a few weeks. We've got the next couple of weeks where Trevor is going to be teaching at, do you want to share real quick where you'll be teaching?
Oh, yeah. I'll be teaching at the John C. Campbell Folk School, Botanical Medicine Manufacturing, where I'm teaching both medicine making, kind of like upper level medicine making techniques to people already trained in it. And then I'm teaching FDA government compliance and good manufacturing practices.
Awesome. So. Well, we've got a real treasure with us, guys, and this Trevor Clark. So thank you, Trevor, for sharing your time with us today. And for everyone out there, may you live long and lucky with strategy.
Yeah. Have a good one, everyone.
Take care.
Here and the numbers beat the game.
He talked to roots and healed his pain.
Data and the dirt, that's the way. Science and the soil.